So yes, here we are, chapter 3. So we're going to start again with the basics of the cell. I'm not going to review the feedback loops again today, but yeah. So this chapter, of course, getting all into the cell membrane. That's where we are in lab. A lot of the transport things that we're doing, and we're going to be getting into the organelles of the cell, of course. And that's it. We'll finish out with the nucleus, and that'll end our Unit 1 information for lecture. For lab, we still have Lab 4 to go. Covering the cell. Right now we're doing the body fluids, of course, in lab, getting introduced to all the transport mechanisms. So I have my last lab today, and I already have a lab video posted if you haven't seen that already. So definitely don't want to miss that. It's all the same stuff, but we have an experiment in lab that's very good with sucrose. So play with that. That's one of the best ways to learn it. And of course, you'll get specific questions on that. And I think I did send everyone a video link to review the solute concentrations and compartments and such like that. And I sent a video link for the chemistry stuff. So they're each, I don't know, about 8 minutes or so. Check out those. Real brief stuff, give you some good information. Okay, so in our hierarchy, excuse me, we're on the cell level. Please don't forget this question. There's no reason to get this wrong on the test. It's all basic, all same thing we've done. It is the basic structural functional unit of life. All these other bullets have to do with the cell theory itself. Of course, all organisms are composed of cells, and all the function of the overall organism depends on the activity of those individual cells, being the metabolism itself, the anabolism, catabolism that we've already done. So all the specifics here of the cell theory are listed. And of course, cells come from pre-existing cells through DNA replication to hold the continuity of the chromosomal number of 46, which is represented as 2N. We'll learn about that in chapter 4. Each N represents the DNA copy you get from your mother and then the father, so that's why it's 2N, representing total of a 46 chromosome cell. All human cells are 46 chromosomes with the exception of the sperm and egg cells. Those are each 23. So yeah, we'll cover more of that in chapter 4. Our fluid compartments, again, just understand you're familiar with the word compartment. A compartment in real physiology indicates the area inside the cell versus the area outside the cell, which is called intracellular or extracellular. In lab, we consider compartments anything we want. If you got 2 beakers on the table and you're asked to compare those 2 beakers, the solute concentration, well, guess what? That's 2 compartments, 2 different compartments. Or if you got 1 beaker and you got a bag, like this experiment lab, in the beaker, well, inside that bag is 1 compartment, outside that bag is the other compartment. It's supposed to signify the intracellular and extracellular environments, but in lab, that's how we do it. Or if you have 1 beaker, and in the middle of that beaker is some kind of membrane, some kind of separating wall to separate those 2 environments in that 1 beaker, that could also be considered 2 compartments. So what we're doing when we talk about solute concentration and solvent, of course, and then movement of water across that membrane, whether it's fake or real, that's describing water moving from an area of its own gradient of higher concentration to an area of lower concentration of water. But on the flip side of that logic is water will always be chasing the higher solute area. So that's why it's real important on this slide you understand the difference between solute and solvent. If we give you a example of a beaker, let's say again on a table, and it's labeled 10% sucrose or 30% sucrose, doesn't matter what the number is, we're going to ask you, what is the solute? And we're expecting you to say sucrose because it's labeled on the beaker, right? Or if it says a 15% solution of sodium chloride, what's the solute? It is sodium chloride. It's just that simple. If we ask you the percentage of the solute or the concentration of the solute, then you can name that number of 15%, 30, 10, whatever it might be. So make sure you're answering the right answer to the to the correct question. So once again, on a 15% sucrose solution, what's the solute? What would that be?
The sucrose.
The solute would be sucrose. Yeah. And what would be the concentration of that sucrose? 15%. Okay, so now if we take that one compartment, we're not comparing different compartments yet, just solution in a beaker all by itself, right? That's just one compartment. What if I say if there's 15% sucrose in that beaker, how much water is in that beaker? What's the percentage? Each compartment has to equal 100%.
50.
So if it's 15 sucrose, it would have to be 85% water. So it's just that easy. And we'll see some examples as we go. Certainly it's all over lab. But yeah, when we talk extracellular in lab, of course, it's just going to be outside the bag like that. But in real physiology in the body, two types of extracellular fluid. Interstitial, which would be the fluid between all cells in the human body, all around the body, I should say, whereas plasma is just in the bloodstream. So plasma is a specific extracellular fluid just in the bloodstream, of course, because it's found around blood cells. That's it. So the water in the blood outside the red blood cells, white blood cells, we don't call that interstitial, we call that plasma. And comparing plasma and interstitial, they're roughly the same. They're all water basically with a bunch of proteins, but the plasma has more proteins. So that is a signifying difference. Plasma has more proteins. If you're asked what's the difference between the IF and the plasma, well, of course, the difference is the place in the body. Plasma is only in the blood. But the other difference is that plasma has much more protein. It has all those carrier proteins to help with our bloodstream, to carry the lipid-soluble particles through. So yeah, know those simple terms. It can be messed up if you don't know the simplicity of it. And know that most of the water, as you see, is going to be in 2/3 inside the cell. That's another important note. 2/3 of volume of water in your body inside the cell. That's a big deal. 3 main parts as we did before. Another question. It's a very common question. What are the 3 main parts? Out of the dozen parts that we're going to study, know these 3 main parts. And a little bit about each. Of course, we know the plasma membrane, cell membrane has many names. It is a boundary to the cell. It surrounds it, provides transport in and out of the cell. I should say controls transport is a better way to put it. And itself is made of proteins and lipids, and making up the glycocalyx on the surface, all the separate membrane surface proteins that we've seen. In the cytoplasm, we got organelles, some cytoskeletal fibers, which are, again, proteins, making up the shape of the cell. And then the cytosol being the actual fluid inside the cytoplasm, mainly water, some ions and proteins, of course. In the nucleus is the DNA, easy stuff. I think this is like, what, 7th grade information? So I'm hopefully not saying much that's new to you. Maybe the word chromatin is new to you. Just understand that's just a condensed form of the chromosomes wrapped up with some histones there, but it's still DNA. This is what all the information we need to make proteins. And my gosh, once we get to chapter 4 and you see what it takes to make a protein, if you haven't already, fallen to your knees in absolute humility and gratitude at the intelligence of your human body, you certainly will in chapter 4. It's some beautiful science going on there to build a protein, and of course, even to replicate that whole cell, duplicating the nucleus. So just a little preview, when we talk about mitosis, that's what we're doing. Yeah, we're splitting the cell into 2 cells, right? But what's really happening in mitosis is we're duplicating the contents of the nucleus. So whereas we get 46 chromosomes in the nucleus, for a moment in time, we have the equivalent of 92 chromosomes, and then we split that cell mitotically, splitting the cytoplasm and the nucleus, and that's how we end up with 46 and 46 with 2 different cells. That's that continuity that I was talking about. So that nucleus is a big deal, big deal. All the proteins that we expect to have in the body, and they're all over, thousands of them, all built from the information in that DNA. It's just that for building a protein, we use something else called an RNA strand. We copy the DNA with the RNA. The RNA is the copy. That's a temporary copy. And then we take a ribosome and attach it to that RNA, and it scans it like a little fax machine. And how it scans it is going to scan the sequence of those the sequence of the bases that we see in the nucleus on the DNA, and it transmits the base language to an amino acid language. So we got a translator. So that's the kind of stuff we'll see in protein synthesis. It's a beautiful process, very complicated, but in its simplicity, what I just explained, we copy, right? And then we translate. Transcription means to copy, translate means to translate. Those are the two main stages of protein synthesis. So I like to mention it here because the way I like to teach is I like to mention things several times. So you hear it now, maybe you got a piece of it, maybe you didn't, but when you hear it again, you'll say, oh yeah, I've heard that before. That's starting to make a little sense. Well, the nucleus is surrounded by the nuclear envelope, which is just like our cell membrane, actually. It's just around the nucleus, and they call it a nuclear envelope. And here's our cell membrane and its beauty. You can see the phospholipids arranged in a bilayer, one layer there, one layer there. That's why they call it a phospholipid bilayer. It's also known as a fluid mosaic model because along with the phospholipids, you got cholesterol units giving strength and flexibility to the cell membrane. You've got tons of membrane proteins all over the place, whether they're integral, where they pass all the way through possibly, or even halfway through, or maybe they're just on the surface. This would be a cell surface antigen. Lots of cell surface antigen. This picture doesn't do it justice. We mentioned some of the most important cell surface antigens, of course, are going to be those that identify that cell as being part of you. So that when your immune cells scan your body, as they do every day, every second, those immune cells are going to recognize the specific arrangement of this glycocalyx antenna system here. And it's going to say, oh, wow, that's a cell that does belong here. That's what we call self. Whereas it scans another cell, a bacteria or virus, and it's going to say that doesn't belong in the body. That's non-self. Let me attack it. So yeah, those immune cells have to learn what's called immunocompetence to get that skill. And it's going to know your ID of your cell, which is the glycocalyx. So when that word comes up, have some high respect for that arrangement of cell surface protein. It's all very, very cool. Very cool. And where do these proteins come from, by the way? You tell me. Where are they made? Or how are they made? We just mentioned it. They are made from the information in the DNA, right? And then the cell knows how to get those things arranged in the membrane or out of the cell to go somewhere else. So some beautiful physiology going on there. But nevertheless, it's called a mosaic because you got lots of things put together in one place. In this slide, we get a little glimpse of the cytoplasm where we're seeing those cytoskeletal fibers. Very, very cool stuff, giving the shape of the cell. Here's that other picture we saw last time. Last time I mentioned it was an apical surface at the top, basal surface at the bottom. Get used to those terms. We'll see that later in the tissue chapter. Apical at the top, where it communicates with another environment. So this might be the respiratory tract where the air and mucus is passing through, right? Or if it's the digestive tract, it'd be where the food is passing through at the apical surface. At the basal surface is just anchored to connective tissue that's going to be underneath. Different types of cells here. We can see all shapes and sizes. We'll cover this more when we do the tissue chapter, of course, but you can just definitely respect that cells are very, they got their own thing going on in each tissue. They're a little bit different. For that glycocalyx, represented cell-to-cell recognition and binding, of course, for certain receptors. Lots of things can happen at the cell surface. Real important. So these are all the players making up that cell membrane, the phospholipids, as we know. And oh my goodness, if you haven't already, please know what a phospholipid is. This is what makes the cell membrane. So we've got two nonpolar tails that are fatty acids and one phosphate head that's going to be polar. So two nonpolar tails and a polar head. That's what makes a phospholipid. Get that straight, please. One head, two tails. The tails are hydrophobic or creating a hydrophobic region since they're nonpolar, right? And then the heads, of course, are polar, so they make a hydrophilic region, which is a lot smaller when you look at the thickness of that cell membrane. So let's check that out real fast. go back to that slide. Here's the phosphate head and the fatty acid tails. So yeah, the thickness, the overall thickness is the nonpolar region, isn't it? So that's why we say that the cell membrane will behave in a nonpolar fashion, because it's, at least the thickness of it is mostly nonpolar there, which is going to dictate how molecules or ions are going to pass through. Those that are nonpolar will have no problem passing through. Those that are polar will have to, or anything with a charge, I should say, will have to use one of these membrane proteins to get through. And that's going to be known as a channel or a carrier. So if you heard that in lab, hopefully it's stuck. Hopefully you got it now. Of all the membrane proteins, some are used as receptors. Right here, you can see it's waiting for a binding site of something. This is for cell-to-cell recognition, so to speak, because it has this little antenna going on. This is an example of a membrane protein channel. So if I say that word 2,000 times today, hopefully you have a picture in your head to go with it. This is where water-soluble molecules will pass through, whether it be a polar molecule like a sugar or an amino acid, or even water. Water's polar, of course, so it has its own little channel, a dedicated channel, a smaller channel. And even ions. Ions have a charge, so they need a channel. All of those things I just mentioned, by the way, water, sugar, amino acid, ions, they're all water-soluble compounds, aren't they? So water-soluble compounds will need help to get through. They need this protein. Lipid-soluble compounds, they don't need help. They go right on through. So that's our two types of diffusion, simple and facilitated. Simple is going right on through, no help. Facilitated means you need help. That's what facilitate means. So if you got the real true understanding of those two forms of diffusion, you're golden. That's beautiful. I think if you understand it in the words that we just stated, then it's yours forever. And you don't have to memorize it, you don't have to study it, it just kind of makes sense. You know you're sitting pretty when things just make sense, because then your brain feels like you don't have to really work hard to study it. You can study the details, I guess, the definitions, but at least conceptually you understand the difference between the words simple versus facilitated. Yeah, so we'll hit that in a little bit later and in detail. So yeah, all this important for the integrity of the cell membrane to withstand all the rigors it has in that tissue. Here's a good, a nice summary slide of the glycocalyx. Lots of things going on. So beautiful to me how the body knows how to build all this out of the crap that we put in it, right? Doritos, donuts, and it still does it somehow. Amazing. But of all this stuff, recognize cell-to-cell recognition and immune system function right here. Those are the big players that we'd like you to know. Cell adhesion, which we're about to get to. So many things. In summary, the three main functions here: boundaries, as we mentioned, interactions with other cells, as we just mentioned with the glycocalyx, and then passage, which is our transport. So that's where we're headed. Something else about membrane proteins, they have many different kinds of those, of course, like we said. Those that can be controlled with voltage, the electricity, the nervous system stimulating a cell. Those that have to bind or that are bound with chemical messengers. That would be what's called a ligand. So when a hormone comes to a cell and needs to bind, basically what I like to call knocking at the door to get permission to come in, which, by the way, let me ask you, and hopefully I can still hear you guys. Out of what we've already learned, let's jump to AMP2 real fast. Let's fly to the future. If I told you there's two families of hormones, protein-based hormones or steroid-based hormones, from what we know already about those two words, recognizing which one's polar, which one's nonpolar, which type of hormone you think would have free access coming into the cell? A steroid hormone, which is nonpolar, or a protein hormone?
Protein hormone.
Think about it. If a nonpolar molecule presents itself to the cell, does it have free access into the cell with a nonpolar membrane? if we go back to our little picture here, where was it? If we said that most of the cell membrane is nonpolar because of the tails, and I told you because of that, nonpolar molecules have no problem going through. If a steroid hormone is nonpolar, what do you think?
A steroid.
There you go. Steroid hormones would go right on through. They don't need to knock at the door and get permission. Their receptors inside, so they go right on, they slip right. Remember I said simply slippery lipids, steroids, and gases? They slip right on through and they find their binding, their receptor inside the cell. Steroid hormones do. Protein hormones, which are basically all the other hormones except for, testosterone, estrogen, those are all steroid hormones. Protein hormones are going to have to bind. They're going to knock at the door, and then they get other things that happen that carry their message inside. So different than a membrane protein, we're talking about hormone binding. But this might be an example of a hormone, a protein hormone having to bind, which I'm talking about ligand. That would be a ligand gated in a way. So yeah, that's a little bit something different, a bigger story. It's not a channel though, but it is a ligand. It's a binding. That's what I'm trying to say. When something binds in the body, we're going to see this through and through. Anytime something binds to something else, some kind of chemical change is going to happen, right? Whether it's an enzyme converting that reaction to go faster, or whether that binding opens up a channel, allowing other chemical messengers to come inside. Lots of things happen. With the nervous system affecting a cell, yeah, it's going to make that channel open for sure. And then we have mechanically gated cells that channels that respond to the physical stress of the cell. Okay, that's just more about the membrane proteins being identity markers and adhesion we're going to get to in just a bit. So of all the functions of a membrane protein, this just kind of lists a great slide showing you what they can do. We're mostly concerned here. We're going to cover a cell adhesion molecule, of course, some of the proteins and such that do that. Identity markers, channels, whether they're gated or not, either way, just please know what a channel is. We've covered an enzyme, of course, and with a receptor, that just has to do when something binds to make it open or not, or respond. This is exactly what medications do, if I didn't mention that before. Any medication that you take is going to take up a binding spot of something that would normally be, a natural reaction in the body, but it's going to take that binding site up and make that cell behave differently, basically blocking that binding site possibly. So yeah, medications are like that. They kind of trick the cell. So the integral proteins versus peripheral. Peripheral just means they're on the surface, whereas integral means they penetrate inside the membrane. So these are all, whoops, sorry about that. These are all integral proteins because they're integrated in the cell membrane. If they were peripheral, there'd just be a little bump on the surface, inside or outside. So there's an example of a peripheral, and this will be an integral coming all the way through. Another peripheral down there. Of course, it's not fair because you can't really tell if that's going all the way through or not. But if you get a picture like that, and that's all they're showing you, then go with peripheral. Yeah, whereas they're showing you something passing all the way through, go with integral. I realize that can be a little misleading because you're not even able to see if that one goes through or not, since it's in a different level, different plane. Okay. There we go. So where are the hydrophilic and hydrophobic regions of the cell membrane? You tell me. All these nonpolar fatty acid tails, would that be philic or phobic?
Phobic.
Phobic, right. The phosphate heads are that represent the hydrophilic region. Perfect. And that's going to be something that you actually get tested on in lab, probably on an illustration like this. Okay, more of the same. Integral proteins, peripheral proteins, glycocalyx at the top, cytoskeletal fibers giving you the shape of the cell on the inside of the cell. There you go. So one simple thing, I mean, this slide is labeled, thankfully, the cytoplasm being the inside and the extracellular being the outside. But if you didn't have those markers of extracellular, intracellular here, would you be able to recognize which part is the inside of the cell versus the outside? The way to do it is those cytoskeletal fibers give you the shape on the inside of the cell, and you always have this little, what I like to call antenna system on the outside, giving you the glycocalyx. That's how cells communicate to each other. You know, cells are so cool. When you see more of this in the immune system chapter, let me give you an example. When one cell is infected with a virus, right, it's going to create these certain little proteins called interferons. And those interferons are going to be released on the fly, right? Since it knows it's infected, that interferons are going to be released out of that infected cell, warning other neighboring cells, hey, I'm infected, right? And guess what? Not only does it warn them that it's infected, it has small pieces of that virus inside of it so that it can teach the healthier cells, the ones that are not infected, it can give a little data on that virus so those other cells can start building some information on how to fight that virus. And the infected cell, unfortunately, you know, kind of sacrifices itself in doing that. It dies. But the healthier cells now have data on that virus. And yeah, cool stuff. Your body is amazing. When you get a patient that's all diseased up, got all kinds of diagnoses left and right, you know, you can look at that patient two ways. You can look at it as, oh my God, this is a difficult case. Or you can start to think, wow, I learned so much beautiful physiology in school. I know this person has a chance because the body wants to survive. Let me just help facilitate that chance. So be in the mindset of helping the patient rather than labeling the patient with all kinds of words and bad things all the time. You need to diagnose, of course, that's how you get paid. No doubt. But yeah, still have the idea that that body is amazing and it's trying to do its best. So be positive with those patients. They need it. All right, some specific cell extensions. So we're going to get into the adhesion things in just a bit. For cell extensions, I did not cover this last time. We have certain, you know, specific cell extensions, not that many really. The cilia being one of them. Flagella being a specific one only in the sperm cell. That's it. There's nowhere else in the body where you find a flagella, not in the human body. But cilia we're going to see in different areas. We're going to see this in the respiratory tract. Another one is microvilli. We'll see that in the digestive tract. But when you think of cilia, it's just a little more on the flagella. I'm not really going to say much about that. It's a tail, helps the sperm cell swim. Nothing really left to say about that. Here's the cilia. Don't they look like little hairs? Well, they may be described as hairs on top of the cell, but they're not hair. They are not hair at all. You need to think of these as, you know, I'll tell you what, do this. Everybody look at their hand, right? Look at your hand. Look at those 5 fingers coming off that hand. These fingers were not just placed on your hand, were they? They're basically coming out of the hand. That's how you need to think of a cilia. A cilia is not just something poked in there like a hair, you know. It is an extension of the cell membrane. So in this picture right here, I'd like to demonstrate this cilia is really just that cell membrane swooping up and swooping down again. It looks like a finger, right? But it's cell membrane stretching out and then coming back again. So yeah, if you were to magnify this picture, you would see a little phospholipid bilayer around this each cilia, right? That is the cell membrane stretching up, stretching down, up and down, up and down, up and down. That's what they do. Okay, and in this case, they help push mucus in one direction. So that's why you have a natural state in your respiratory tract of mucus being pushed up to clear out the lungs. So for those that have smoked for a long, long, long time and they have this regular cough, that's because the nicotine paralyzes those cilia, and the cilia can't do their job. They can't flow that mucus, you know, keep it coming up, clearing the lungs. So that's why you got to cough, because guess what? That mucus gets kind of thick, especially if you're dehydrated. Yeah. So that's what cilia do. They push mucus across the surface. Very, very important. And a lab example we're going to get next week describes how some of that is helped with chloride pumps. In the case of cystic fibrosis, now you tell me, when I say chloride pump, do you know what that is? First of all, what does it pump? Does it pump chloride ions or sodium ions?
Chloride.
Chloride ions, perfect. So what are these pumps made of? Carbs or proteins? What are these membrane channels and pumps made of? We just mentioned it.
Protein.
They're all made of protein, right? So yeah, this is a DNA issue. This is a genetic disease where that person with cystic fibrosis, they're not producing those channels, right? They're not producing those chloride pumps, or they just don't work. So if the chloride doesn't get pumped out, the sodium doesn't get pumped out, because sodium likes to follow chloride always, and vice versa. So if the chloride's not pumped out, the sodium's not pumped out, and then the water has nowhere to go, because without a concentration that's different, water will naturally want to follow sodium chloride. Well, if sodium chloride's not moving, water's not going to move. So that makes the mucus thicker. And that's what gets happened. You get these mucus plugs in the respiratory tract mainly. So we'll do that in lab as an experiment, or at least a little clinical talk. Another extension, microvilli. Real tiny. Look at this. They're not as noticeable as the cilia. They're very, very, very tiny. But again, they're small extensions of that cell membrane. Very, very important. This is what's called the brush border. This is what helps intestinal cells here. This is your lining of your stomach and intestines, by the way. This is what helps you absorb food. Definitely. It gives what's called increased surface area. Yeah. So think of your bedroom. If you've got carpet in it, right? If you got a flat piece of carpet going across the floor, right? That does represent kind of microvilli, little carpet fibers coming up. But think about it this way. Think about you folding that carpet from the floor to the ceiling, right? Over and over again across the room. You would end up using a whole lot more carpet doing that. That's what microvilli is. Microvilli increases the surface area of the top. What's the name of the top surface, by the way, of a cell? Is it apical or basal?
Apical.
Apical, right. So is microvilli present at the apical surface or basal surface?
Apical.
Apical.
On the website medcell.med.yale.edu,
Test question. Oh my goodness.
they say microvilli are small
You hear that?
particles that project from the apical
Yes.
surface with most types of epithelial cells, greatly increasing the surface area.
All right. Y'all got Google teaching you now. Hey Google, what's the study of anatomy physiology?
On the website thecompleteuniversityguide.co.uk, they say anatomy and physiology is a line of science concerned with the bodily structure of living things. Do you want a little more context?
Nope. Hey Google, what is the apical surface?
On the website medcell.med.yale.edu, they say epithelia cells are polarized with an apical surface that faces the lumen of a tube or the external environment and a basal surface that attaches to the basement membrane.
Oh, wow. She said it better than me. Wow, we can learn a lot from her. Well, cool. That's another resource. Yeah, apical surface at the top. So test question, is the microvilli at the top or bottom of the cell? It's at the top. Is it the apical or basal? It's at the apical. So that's the best way to learn stuff, is just get it all cleared up like that. Or if you can't, you can just ask Google. You know, the idea that we have Google or Siri, whatever you want to call it, or Alexa to talk to, that was all stuff way back in my day when I was little, only in the movies. That's hilarious that we're like living in the future already. Isn't that cool? The Jetsons. Anybody know the Jetsons? Nope.
Yes.
Wow.
Yes.
There you go. The Jetsons were it, man. That was the coolest of the cool way back. So here's another good shot of microvilli. Check it out. Now you can definitely see the actin filaments that give the strength of these microvilli coming way up and way down. And if you were magnified, you'd see a phospholipid going way up, way down. Beautiful. Actin filaments, protein. Again, proteins are everywhere. So let me ask you, do you need protein in your diet? Absolutely. Oh yeah. Do you need water in your diet to sustain those proteins so they don't get weak and denatured?
Yes.
Oh yeah, lots and lots and lots of water. So, let me just say one more time. When I say that soda is sludge for the body, we're all going to drink soda. We all love it, right? But we all have to admit it is not good for us in any possible way. It doesn't matter if it's diet or whatever. Diet's even worse, actually. But yeah, limit your soda if you can. It's tough. It's part of our culture almost. But do yourself a favor, don't drink soda every day. Break it up at least, maybe every other day and then go to every 3 days. Do not drink soda every day. I know it's tough because it's just part of what we do, part of what gets us through our day. But you'll notice if you can get through those first 2 weeks of difficulty, you'll notice you'll start feeling better. When you're young, you really don't notice those kind of things. So maybe I'm not talking to the right crowd because most of you guys are young, but it still affects your body. It's going to build up. When you get older and you feel aches and pains just out of nowhere for no reason, examine that and say to yourself, well, yeah, I'm not exercising, but what am I putting in my body? If you're eating a bunch of packaged foods and drinking soda, I can tell you that's a lot of where your aches and pains are coming from. And if you're a smoker, you're going to get aches and pains, right? So water clears out the body. Let me just put it that way. Makes everything better. All right, cell junctions, 3 types, tight, desmosome, and gap. Here's a tight junction found in the intestines, keeping those intestinal cells nice and tightly wound together because we don't want our intestines leaking, that's for sure. A plaque linking up 2 cells for strength and durability, like in the skin and the heart. And then a gap junction, sharing electrical connections in the heart, so the heart can beat as one unit. Electricity, when it hits the heart, it goes completely through the heart, even though it just stimulated a few cells. It's shared all the way around the heart. So the heart is known as a syncytium in that way because the heart can beat as one unit, thanks to these special gap junctions that spread the electrical charge around the heart immediately, and in smooth muscle as well. So know the examples for each of those, gap, tight, and desmosomes. All right, I hate that we lost 30 minutes of our day. That really stinks because we could have gotten completely through transport here. But here's the summary functions of the membrane, of course. Now we get into transport, active and passive. This is the same exact information presented in lab. So if you've got it in lab already, then great. It saves us a lot of talking, but we will be going through it. You just get more examples, I guess, in lab, so it's more easily learned that way. Well, we got passive, transport, and active. If you learn passive, then you're going to learn active automatically because it's the opposite, at least the principles. So with passive, it's no energy. It's considered downhill, which means we're moving from an area of high concentration of a solute to an area of low concentration of a solute, or water. High concentration of water to low concentration of water. High solute to low solute. That's downhill. That is a gradient, by the way. If there's no concentration difference across the cell membrane, then you really don't have a gradient. You don't have a drive. You don't have a reason to go somewhere else. So we need solutes to be basically unequal, don't we, on either side of the membrane to influence water to move. And that's what we talk about the term osmolarity and hypertonicity. When solute concentrations are different on one side of the membrane compared to another, that's what builds that natural drive for water to be pulled in that direction. So think of water being drawn or pulled or swayed or influenced, or bribed, if you want to put it that way, whatever word suits you. Water will be influenced by high solute concentration, right? It's going to want to move to where solute is high so it can equilibrate it. That's just the natural tendency of water. It's like, oh, look at that solute over there. Bam, that's where it goes. That's how water behaves. Oh, look at that solute over there. Well, wait a minute. It's a higher solute concentration over there than compared to over there. So I'm going to go here first. It's going to chase the higher concentration, or at least it'll go faster. It's going to chase every concentration, but it's going to go faster on a steeper gradient. So you tell me, without even seeing a picture, what represents a steeper gradient? A 65% solute solution or a 30% solute solution?
30.
The steeper gradient is the 65% solute solution because there's more solute. So it can be tricky when you look at everything on an example, but just saying it out, whatever the higher number is of solute concentration, that's the higher solute, right? And water will chase that faster than the other gradient. So they're both going to go downhill, but you tell me, if you roll down a steep hill, right, versus a very shallow pitched hill, you're going to roll down that steep hill a lot faster, right? If it's going like any skateboarders, anybody rode a half pipe, the vertical, that was part of my past. Yeah, you go pretty fast down that ramp compared to a driveway. You know what I mean? Yeah. I used to love to skateboard. Well, not skateboard, but I used to love to climb on everyone's roof in the neighborhood and then jump off. That was my favorite pastime. If my kids ever did that, my God, I tell you what, I feel old when I say things like that. Because I certainly wouldn't do it now. Hell no. No way. Here's your 4 types of passive transport. And we're right out of time here, but let me just push through these 4. We did them in lab. Simple is going to be for lipid soluble, facilitated for water soluble. And then we have osmosis specifically just for water, and then filtration representing the pressure of water. So in lab, we went through all the details, which molecules move through for simple, which are all going to be nonpolar. There you go. And then we described simple going straight through just like that, no help needed. Whereas facilitated does need help. It helps water soluble molecules move through and ions using carriers or channels. These are membrane proteins that we saw. And there you go. There's an example of a membrane protein helping a solute there, or an ion. There is a carrier being a little more specific on shape. And there's a specific water channel called the aquaporin. So we do these same slides in lab, so you'll probably be good there. Let me end up with this one. The IV fluid, yeah, that would be the saline, right? Isotonic to plasma. Isotonic means it matches the solute concentration. For aquaporin, would an osmosis speed up or slow down with more aquaporins? The nature of osmosis is to move through an aquaporin, so yeah, it would speed up. We want those aquaporins for osmosis to take place. And we'll pick up here next time and get more detailed about osmolarity and hypertonicity. We'll finish this up on Tuesday and get through the organelles as well. All right, guys, any questions? I'll be here for a little bit. Thanks for your patience today. So sorry we lost some time.
Dr.